So, besides the common sense that there is in having pruning techniques that respect the sap flow, there is also an important aspect of Sandro's point of view. It's about reserve preservation. And actually, since now we are going to dive into the leaf area period, it's important to understand, particularly in the context of warming temperature, that management of vine trunk reserves is key, particularly to initiate bud break. Typically, when you look in the literature, it's expected that until the shoots reaches 6 leaves, most of the bud break will rely upon vine's intrinsic ability to tap onto the starch and other carbohydrates that have been accumulated. So as temperature gets warmer, starting to think pruning also as a function of reserve management makes a lot of sense. And this actually is a little bit what I'm going to touch base upon by focusing on comparing growth rate. The goal in the next few slides is going to compare a vintage such as 2014, dry winter, meaning lower nitrogen uptake, with 2015, since we know that 2015 benefited from more soil moisture and higher nitrogen uptake. So why growth rate is important from a winemaking standpoint? Well, first of all, growth rate happens very fast from 0 to 400 growing degree days after bud break. And the speed at which the plant grows directly impacts the piping system of the plant. The hydraulic properties of the plant are derived from growth rate. Then another thing, it's exquisitely sensitive to stresses, whether those stresses are induced by nutrient deficits, water deficits, or disease, like trunk diseases. And last, the growth rate, as Sandro was saying, is related to the size of the bunch. So growth rate is related to yield. And it shows very early, so you understand that growth rate has a predictive power. So how do we standardize our study of growth rates to compare years in, years out, how different vintages impose a different footprint on growth rate? Well, we have a very simple idea. This rectangle symbolizes the leaf area that's going to be exposed to sunlight. And we know that by 400 growing degree days, most of your canopy should have taken place into this rectangle shape that symbolizes the canopy of the vine. So by tracking at key stages of leaf area development where we are, we can tell whether or not growth rate is lagging behind or is ahead of its time, considering time is expressed in thermal time. So I have listed over the last 2 years, and I have used 2011 as a baseline since things happened remarkably slowly in 2011. And you see that 2015, as expected from thermal time accumulation, is a little bit ahead. Not so much. We roughly had gained 8 days by the time we reached 400 growing degree days. So the take home is that by May 15, we were expecting most of the leaf area development to have reached its peak. But now we can look for a specific period at how quickly shoot elongated. And this is what we've done. So here each line is a statistical analysis of more than 200 blocks where we have days in, days out, monitor the rate of shoot elongation and then plotted it in order to compare things according to thermal time instead of civil calendar. And what pops up out of this trend comparison is that even if we were taking into account thermal time, 2015 showed a leaf elongation rate that happened much faster than 2014. And based on the first observations we made earlier, we know that nitrogen uptake, greater nitrogen uptake combined with denser root network is probably at the root of this plant response, which means several things. It means that maximal leaf area exposed to sunlight will be reached earlier, so under warmer and longer days, which could enhance water deficit severity later on. And it's not only true with Cabernet Sauvignon, it's true with Shiraz, it's true with Merlot, with a different rate. So for instance, with a Shiraz, we've observed 25% increase in the speed of shoot elongation. With Merlot, barely 10%. With Cabernet, we are almost at 40% faster. So different varietals have different response to these environmental settings. So now, what are the questions we can ask? Well, higher root uptake in terms of nitrogen and faster shoot elongations have been observed. We expect that it should have set the stage for a greater yield due to the faster speed of shoot elongation. However, it's not a secret. In 2015, despite a bunch number per shoot that was rather unaffected, we had a low number of berries per bunches. So what in the vintage conditions may explain these tendencies that we've observed to see low yield component on the berry size number per cluster. So I looked at mean temperature around bloom because it was suspected that lower than average temperature should have been somehow responsible for the lower berry number per cluster. However, even if it is true that temperature during the key stages stretching from bloom to set were relatively low compared to 2014 or 2013, it's not exceptionally low compared to 2011. As a matter of fact, in 2011, still comparing in terms of growing degree days, you see that during the first 50 growing degree days after bloom, 300 to 350, temperature were a few degrees lower in 2011. However, 2011 is not remembered as a low yield year. And then for the later varietals in terms of set, 350 growing degree days, you see that both vintages were still above 15 Celsius. And 15 Celsius is the temperature threshold below which we expect berry sets to be negatively impaired. So nothing is screaming at temperature being the main culprit to explain why we had such a low berry number per bunch. It may have contributed, but it doesn't explain the whole trend. Then I looked at rain. And again, sure, we had some rain at set, and sure, it may have affected negatively in some places the amount of berry number, but it's nothing compared to 2011. So again, an explanation that may contribute to explain a lower yield, but it doesn't give us a rounded picture. So, based on that, what could be other factors responsible to explain why we had such a poor set? And that's where it's very interesting to look at the footprint and carryover effect of one vintage up onto the next. And there is a growing body of literature. I quoted two authors here, Holzapfel in Australia and Gilpart in France, and they have shown that there is a direct carryover effect from year 1 onto year 2 caused by deficits, whether they are water stress, whether they are nitrogen stressed, onto year 2. So it's tricky because you may not see that a year is actually setting up the stage for a lower yield because in 2014 we were not specifically marked by a decline in the yield. However, we had a poor nitrogen uptake in 2014, which I will discuss soon, but just to put things into perspective, it makes sense to link nitrogen uptake dynamics season 1 with season 2 yield. And what do we know about nitrogen dynamics? Well, nitrogen uptake is not stationary. As a matter of fact, it is usually variable. Here you see how the footprint of different vintages is affecting the time profile of nitrogen uptake. And really the take home to get from these slides where you see we had 7 to 8 points taken from split flower stage to bloom up until post-maturity, you see that the rate of nitrogen uptake is the highest before bloom, is the second higher at bloom, and then it's a very fast decline. It means that when you try to correct nitrogen level by applying fertilization late season, it is less efficient than early season. So nitrogen uptake is at its highest early season, it declines with time, and it reflects the vintage effects. So now that we have taken that step back, what were the conditions of nitrogen dynamics in 2014? Well, on the 100 plus sites where we were actually monitoring nitrogen early season, what was screamingly high was an extremely low compared to other years uptake of nitrogen. And we know why. We had for sure high temperature, but we were lacking soil moisture content during the winter 2014. And the carryover effect was that during the 2014 season, we had a poorer nitrogen uptake, and the price to pay is a lower berry number per bunch in 2015. Or at least that's an hypothesis that we can bring forward to explain also why it's really possible to link the low yeast assimilable nitrogen that was contained into the must with its carryover effect in 2015. So that's the third key take-home message. When we compare side to side a fast elongation rate in 2015, which should lead the way for a higher yield, with respect to the low nitrogen uptake in 2014, it was kind of hard to weigh which of those two components was going to drive the yield. Well, in fact, we've observed that the heaviest weight was carried over by the low nitrogen uptake in 2014, in spite of having had faster shoot elongation rate. So, carryover effect of nitrogen uptake is not the only component of yield definition. Actually, the level of vine health is very important. Sandro spoke about how to minimize the onset of trunk disease, and soon Dario Cantu is going to explain how the research is leading the way and improving knowledge in that field. But before jumping into that, I simply wanted to show you that the visual symptoms of a plant that's infected are not always the best clue to characterize what's happening inside the vine. And we understand through the cone of desiccation how it could affect the trajectory of the sap. Here I wanted to share with you a water deficit index profile taken on two types of plants. Those plants, whether they are blue or gray, looked perfectly normal. There were no foliar symptoms. However, inside the plant, because of infection caused by grapevine trunk diseases, you see that from middle August onward, we have a major decline in the amount of water use, which directly drive how quickly sugar is going to be downloaded into the berries. So I thought this slide was going to be a nice segue to introduce our next speaker, Dario, who is going to share with us basically his insight and results in grapevine trunk disease. Oh, yeah.
Okay. Good morning, everybody, and thanks the organizer for giving me the opportunity to present here. So in the next 15 minutes, I will give you an update of what is our research and development in improving the current diagnostics tools for trunk diseases. So I will not give you an update on the research on trunk diseases, but I'm going to give you an update on how we can look for trunk diseases, how we can diagnose for trunk diseases in the grapevines. So my name is Dario Cantu. I've been faculty at UC Davis since 2012. I'm also Italian, so this is a very Italian session. But I was adopted by California in 2012, where I've been faculty since. So just to refresh something about grapevine trunk pathogens. They're fungi. They can be ascomycetes or basidiomycetes, so make mushrooms or not make mushrooms. As Alessandro pointed out, pruning wounds are an important entry point for pathogen infection. Some pathogen may come from propagation material, so they may not enter through pruning wounds, but maybe just already present in the vines as we plant them. As they invade the wood tissue, they damage the vascular tissue, as seen here with a typical V-shaped canker in a cross-section of an arm, or as seen here as this necrotic streaking of the vascular tissue in a longitudinal section of the base of the plant. So degradation of cellulose, degradation of lignin, and damage in the vascular tissue. As they spread into the vine, these fungi secrete toxins which can cause now symptoms very far away from the point of infection. So as in case of ESCA, we find measles in the berries, red berries or white berries, both affected by measles. Tiger stripe symptoms, also caused by ESCA, or you're probably very familiar with this type of symptoms, Eutypa dieback with early in spring shoots that fail to grow regularly, cup-shaped leaves, necrotic at the edges, and very short internoded shoots. Okay, so this is in one slide all you should know about trunk diseases for my talk. Of course, as trunk diseases spread into the vine, they cause damage to the vascular tissue that can end up with arms position, which may fail to develop in spring, or some young vine decline issues in case of drought stress or heat stress in combination with pathogen infections. Okay, so a complexity that comes from the last 20 years of studying this disease is that we call it Eutypa, we call it ESCA, but there are so many different species of fungi that call these diseases that the list grows every year, which makes us, which makes diagnosis of these diseases kind of hard. So how do we do this currently? So we take a disease plant, a canker, we make little pieces, we plate it on a petri dish, we allow it to grow, so this petri dish contains now a nutrient which feeds the fungi present in the wood. The fungi will spread, we collect single colonies, we plate on new plate, we do several purification steps, and then we look at the morphology. We look at the colony morphology, we look at the morphology of the spores, and we say, okay, the pathogen that plant is infected of is this species or another species. This requires very skilled mycologists with good eyes for identification of fungi. Current technologies allow us to, once you isolate the fungus, extract its DNA, run a PCR test, and refer to a database and tell which species is actually in your grapevine. Still, both approaches require you to go through a plating step. Okay? And this is laborious. It depends on culturing, so both technology depends on culture, which is a problem because we have a species bias. We know that some fungi, particularly the ESCA pathogens, can grow very quickly throughout the plant. We put them on a petri dish and they grow super slowly. So once we put the sample on a petri dish, now we are favoring the growth of all those pathogens that can actually grow better on petri dish, which does not mean that are the best one growing inside your grapevine. Okay, so we depend on culturing, that's a species bias. Of course, this requires hands-on time, both for culturing and purification of the colonies, DNA extraction, and so forth. And of course, we are looking at one species at a time because we go through purification. We cannot run DNA tests on a mix of cultures. We have to look at one species at a time. So typically, this type of diagnostic takes time and returns you one or two candidate species as the pathogens in the vine. We are proposing to use next-generation sequencing technologies, so-called deep sequencing technologies, to avoid the culturing step. So we take now directly DNA from a canker, process it in a way that allow us to have markers for each of the species present in the plant, and we end up with a figure that tells us the different species present in the canker of these fungi and the relative abundances. So we're adding two things here, three things. We can detect them in planta without going through culturing, so no species bias. We are looking at multiple species at the same time, and we can quantify them. Okay, so 3 advantages in one shot. And why we can do this? We can do this because our capability of sequencing DNA has increased dramatically over the recent years. When the human genome was announced, it took about 10 years and $3 billion. Okay, thanks to the Human Genome Program, now the cost of sequencing went dramatically down. This is on a logarithmic scale, showing the cost of sequencing of a human genome going down from $1 billion almost to what it is a current price of $1,000 genome. Okay, so dramatic improvement in the technologies that we can use that result in a dramatic reduction in the cost associated in using those technologies. And we call them collectively as next generation sequencing technologies. And as the time passes, every 6 months, we basically reduce by half the cost of sequencing, and we increase 2-4 times the amount of DNA we generate in the unit of time. So these are the good news. Why we think that next-gen sequencing and deep sequencing should be applied to diagnostics in the vineyard is because it's rapid. So we generate millions of sequences in an overnight run. The smallest machine, a $100,000 machine, produces 20 million sequences, 20 million IDs of fungi, for example, in an overnight run, and that will cost only $1,000. So it's efficient. It's efficient because when we have a mixed environmental sample, it could be an infected grapevine, when we sequence it, we sequence all the fungi associated to it, all the bacteria, potentially DNA viruses or RNA viruses, depending what we're looking for. And it's sensitive. It's sensitive for 2 reasons. One is an open system, so it allows you to discover something that you don't think is in your plant, so it's not biased by any preconcept of what may be there, and can detect pathogens at very low titer. Okay? So in the past 4 years, we have developed the information that is necessary for us to start developing these technologies. So we have sequenced several genomes. Probably you cannot read them all, but just going through, we sequenced the genome of powdery mildew, of course not a trunk disease organism, but we sequenced the Otypa dieback genome, some of the Botrosphaeria canker genomes, Phomopsis dieback species, and so forth. So we started creating all the information necessary to start applying the technologies in trunk disease diagnostics. And we've been playing with a very recent technology that allows us quickly to have the complete map of a new disease-causing organism in very short amount of time. Okay? Just some example, but this is more about my research program, so we're not using genomics only for diagnostics. We're using these technologies to study these diseases, how they infect the plant, how they colonize the tissue, how they result in symptoms, and how in turn we can reduce their aggressiveness. But of course, in the last years, we have also looked at how to apply this technology for diagnostics. We did a very simple exercise. We drove throughout California. We collected trunk wood with symptoms of trunk disease, ranging from symptoms of ESCA, Eutypa, with or without leaf symptoms. And we tested the old technologies, and we tested the new technology we're developing. So again, you cannot read very well because it's small, and that's my fault. But here you can see. So we collected, for example, from Kearney, we collected this wood tissue with canker and streaking symptoms. We isolated so traditional diagnostics tools. We isolated a species, and we ran PCR on the isolated species. And you can see that we can always find one, in some cases, 2 species associated to the disease material. Okay, so and we did this for a large number of samples, and the very same samples we plated on Petri dish we use for testing next generation sequencing technologies for diagnostics. And here comes the good news. So this is the easiest approach. We just amplify with a PCR reaction the hypervariable region of the ribosomal RNA, again, from DNA extracted straight from the plant without culturing, and we sequence this region. The genetic diversity in that region tell us which species is present in the tissue. Okay, so now you're going to see 2 slides with this type of figure, so better I explain this. Each bar represent a sample. Different colors represent different species of the pathogens. So this blue one is Diplodia seriata, the Aporta ampelina will be the dark green one, the ESCA pathogen, the yellow one, and the green one, and so forth. So now for each sample, we can detect, as you can see here, multiple species. The asterisk refers to the species that was also identified by traditional methods. And I think here is even better to explain how good this technology is. So if we just look, for example, at column 2. Okay, column 2, plating and PCR detected these two species. Okay, these are pathogen species. So traditional methods actually identify pathogen species. However, failed to identify the most abundant pathogen that was present in the sample, this yellow pathogen, which is the Togninia minima pathogen, one of the pathogens of ESCA, so a disease-causing organism, which does not grow well on Petri dish. So by sequencing directly DNA from the plant, we could identify and detect and identify multiple species at the same time. It's easier in the case of Eutypa infections. Eutypa is a very strong pathogen and a good opportunistic pathogen, and it always represents the majority of the tissue. So let's say that if you have the typical Eutypa symptoms, it's very likely that Eutypa will be the pathogen associated to it. If you have cankers and you don't have any other symptoms pointing to a particular disease, this type of technology can tell you which organism is present, and once we know what organism is present, we'll know something about epidemiology, we will know something about his behavior throughout the year, when to prune, when not to prune, and what type of paste to apply when we prune the vines. Okay? Good correlation between technologies. So this is a cross-validation. Each one of them is independent technology, but they all agree in the number of species we identify and the amount of species we identify. Something interesting we can start looking at is how active are these pathogens. When we sequence DNA, we look at abundance of the pathogen. When we look at RNA, we look at their activities. So you can see that most of the time, activity and abundance correlate, of course. In some cases, activity and abundance don't correlate, suggesting these guys are very abundant in tissue but not that metabolically active, which is also another additional information which we can gain from this type of technology. And this is the future, but I see this coming fairly quickly. So when we sequence RNA, I said we sequence the activity of the fungus. What is the activity of the fungus? Is cell wall degradation, cellulose decomposition, lignin decomposition, synthesis of toxin. By sequencing the RNA, we can actually look at our grapevine in terms of all the functions that are active in that grapevine in terms of its pathogenicity. So we take a sample and we can say, okay, now Eutypa is causing the symptoms at the shoot level because it's very active in the secondary metabolism, it's producing toxin, and so forth. Other Botosphaera cankers are causing disease, are causing vascular damage. So we take a very, very accurate and detailed scan of what is happening in the plant in terms of its disease. So concluding, unlike traditional methods, by applying this technology, we can detect pathogen directly from the wood without going through plating. We can identify multiple pathogens simultaneously, and we can quantify their relative abundance. It's not anymore a presence-absence type of analysis. We envision this type of technology to be applied to different material, going from preparation material, how clean is the material you plant, and how careful you have to be at planting not to cause disease development at that stage. You have a disease that is killing your plants and you don't know what it is, you can apply this technology to discover the disease-causing organism and hopefully find a solution to it. And we can start looking at how clean are the facilities that are involved in grafting, involved in propagation, and so forth. We know that some of the pathogens come in at different stages of propagation. We can now go and look for the presence of this pathogen in the facilities as well as in the grapevines. So to make this technology accessible to growers, we have teamed up last year with a company, Microtrack. Microtrack has a lot of experience in applying next-gen sequencing to diagnostics, particularly in food facilities. But we see that as an opportunity for us to make the technology available, so have a real impact in the industry. My lab is still working on the optimization of these protocols, and Microtech will help in making it available in the early future. My team, and I should mention that the work is done with the real plant pathologists like Philippe Rolschhausen from UC Riverside and Kendra Baumgarten from USDA ARS. Funding for all this work is by American Vineyard Foundation. And I will be happy to take questions if you have questions. Thank you for the attention. I don't bite.
Well, I have one question. Have you studied activity in response to either nitrogen or water stress?
We are studying the relationship between spread of the pathogen, activity of the pathogen in relationship with drought stress. We see the drought enhance at least the lesion that this pathogen produces enhances them. We don't know the reason, the underlying mechanism of that, but there is a connection between abiotic stresses and biotic stresses. Young vine decline is a typical example where you have the presence of the pathogen by itself is not sufficient to cause young vine decline. As the plants are stressed at planting, drought stress, mineral stress, etc., then they undergo young vine decline. So there is always an interaction between abiotic stress, how the plant is doing in the field, and the symptomatology and development of disease in general.
And in your result, abiotic stress increases abundance or activity?
Right now we know that it increases the symptoms. So if you just look at the size of the cankers that will develop, they are much larger than in absence of stress.
Okay.
We don't know if that is just because the plant cannot defend itself as much, or the pathogen enjoys the drought stress. We don't know yet what side of is.
Thank you.
Yeah. Thank you. Yeah. Listen. Yeah.
My question is, in Europe now, in this moment, they take a lot of attention in the grafting point, and especially the kind of grafting, because the omega seems increase the trunk disease, the development of the trunk disease. With this method, do you think if you check and detect the vineyard mothers before the grafting for the young vines, it's possible to reduce the percentage of trunk disease or not? It's only just to detect.
I think is key, particularly for these diseases for which we don't have a cure, is critical to start as clean as possible. So everything you can do to reduce the presence of the pathogen is key. It's key to reduce it in the soil of the nurseries. It's critical to reduce it in the facilities where grafting is ongoing. Work done in Spain showed that the grafting machine is plenty of spores of this pathogen. So as you do the complementary cuts, scion and rootstock are directly exposed to the pathogen when the wounds are fresh. So you can take swabs of those machines and run through a sequencing pipeline like that, and you can see how clean or dirty it is your grafting machine. So reduce the inoculum, reduce as much as possible the presence, and then reduce as much as possible the condition that promote disease. Stress is one thing, planting stress is one thing, pushing vines to produce very early on in the lifespan is also known to increase the chances of young vine decline, Petri disease, and so forth. Yeah. Thanks.
Thank you, Dario. Yes, thank you, Dario.
Yep. One more. Okay. Sorry.
That's all right.
Back here. Nope.
Sorry. There was a question earlier about in an applied way, about how to cover the pruning wounds, whether it's a physical barrier or if there is a chemical. Do you have an opinion on that topic?
Yeah, I think it's very important. Timing of the pruning and protection of the pruning wounds is critical, I think. We know that disease pressure is always there. Work done by Kendra Baumgarten, USDA shown that spores are always there. When there is rain, spores are there. Even if the vineyard is young and healthy, spores are there. So as you prune a young vine, the risk of disease development is there. Typically, you start caring about it when symptoms start developing, but that's very much too late. So protection, if it's economically feasible, protection of pruning wounds early on, right away, is key in reducing the incidence. We have done some modeling as part of a large USDA project. We did some modeling and shown that if you protect your pruning wounds right away, you don't get disease in your vineyard, because no entry point, no infection. Then the efficiency of the paste and the type of paste, and very much depends on your type of production system, organic versus not organic, and how much time you can spend painting your pruning wounds, or you may invest in spraying this paste, etc. So, but my recommendation would be yes, that's key.
You mentioned that your type increases secondary metabolism. Does that imply higher quality or lower quality?
So I meant its own secondary metabolism. So type produces toxins that reduce the size of the shoot, toxins that kill the tissue in the grapevine. That's the secondary metabolism we're looking at. Not the grapevine secondary metabolism. Sorry, I should have. There is some work done on the interaction between trunk diseases and quality, and it's always reduce the production. It may in the short term may increase some accumulation of secondary metabolite, but overall is negative. I will not. Yeah. Okay. Thank you.
Thank you, Dario.